Welcome back to this next video in the series of lectures on the cell cycle. In the last lecture, we were discussing the events in the G1 phase of the cell cycle. Now the cells, they usually maintain their internal and external physiological states, but in certain conditions like exposure to the UVB light, which is actually from 280 to 315 nanometer, when your DNA is exposed to this particular light, it can damage the DNA. Now, if the cell is maintaining their internal stability, that means when the DNA is damaged, you have to do something about it. Now, when the DNA is damaged in the G1 phase of the cell cycle, to tackle with that particular damage, the p53 protein that is going to play their role. Now this p53, the famous p53 known as the guardian of the genome, that is actually the sensor of the DNA damage. And if the damage is there in the G1 phase of the cell cycle, it is going to halt the cell cycle at the G1 and to tackle with this particular DNA damage. Now how the p53 is going to achieve this? That is how it is going to halt the cell cycle in the G1 phase of the cell cycle when there is DNA damage. This p53 is actually a transcription factor. That means it can activate a lot of genes. In this particular G1 phase, when there is DNA damage, this p53 is going to work as a transcription factor for a protein known as the p21 protein. And with the help of this p21 protein, the p53 is going to halt the cell cycle at the G1 phase so that it can repair the DNA, or if the DNA is not repairable, removes the cell from the system. Now this p21, when you look at the function of this p21, it is actually going to inhibit the activity of the CDK4 by interacting with it, thereby preventing its association with the cyclin D. Now in the previous lecture I told you that this is CDK4 is the cyclin dependent kinase. Now that means if you want the activity of the CDK, you have to attach with it its particular cyclin. Now if you talk about the cyclin and CDKs of the G1, the particular CDK is known as the CDK4, the particular cyclin is known as the cyclin D. Now this p21 is going to interact with the CDK4, and when the p21 interacts with the CDK4 is not able to interact with the cyclin D. Now that means you are not going to get a complex of the cyclin D and the CDK4, and hence you cannot activate the CDK4 when it is bound to the p21 protein. Now when the CDK4 is not activated, what is going to happen? This CDK4, when it interacts with the cyclin D, it gets activated, and if you look at the substrate of the CDK4, that is the retinoblastoma protein. Now initially this retinoblastoma protein is in the hypophosphorylated form, and it is in strong interaction with a transcription factor known as the E2F. Now this E2F transcription factor is very necessary if you want to move from the G1 into the S phase of the cell cycle, and when it is bind to the retinoblastoma protein in its hypophosphorylated form, the E2F transcription factor is not going to release. You actually cannot move from the G1 into the S phase of the cell cycle. Now when the CDK4 that is activated, what it is going to do is it is going to convert the retinoblastoma from the hypophosphorylated form into the hyperphosphorylated form. Now when the CDK4 is not activated, that means you cannot convert the retinoblastoma from the hypophosphorylated into the hyperphosphorylated form. That means you cannot release the E2F from the retinoblastoma protein, and if you cannot release the E2F from the retinoblastoma protein, you cannot move into the S phase of the cell cycle, and you are stuck in the G1 phase of the cell cycle. Now once you have stopped the cell cycle at the G1 phase, this is you can say a diagram showing you the functions of the p53. Now the p53, it deals with the damage in two ways. The first thing is that the p53 is going to arrest the cell cycle, as we just discussed how we are going to arrest the cell cycle in the G1 phase of the cell cycle. This p53 is going to activate the proteins which are involved in the repair of the DNA, and when the DNA is repaired, the cell cycle is going to restart again. Now, if the p53, it's a rest cell cycle, it attempts the DNA repair, but it fails to repair the DNA, the p53 is going for the process of the apoptosis. Then it is going to activate the proteins involved in the process of the apoptosis, and with the help of the apoptosis, it is going to remove the damaged cells from the body. And in this particular mechanism, the p53 is going to ensure the cellular and genetic stability. We'll continue this discussion in the next video.